demonstrated the same result. The results of our study are consistent with the hypothesis that excessive exposure to prenatal androgens, read testosterone, in women with CAH shifts psychosexual differentiate to a point somewhere in between a female typical pattern and a male typical pattern. These women, many, have varying degrees of erotic orientation to women. Now, it's just not the hormone that's involved. In order for the hormone to send its message, it must be captured by the target cell, which then relays the message to the nucleus where the genes interpret the message. The key molecule is a protein, which is a receptor. So testosterone or estrogen or cortisol, in order to make an impact on that cell, have to get into the cell, bind to the receptor molecule, which then permits it to be relocated to the nucleus, where it finds a place to sit down and causes either a gene to be turned on or a gene to be turned off. There's a condition where there's a genetic mutation in the receptor for testosterone. So think about the consequences for a male. This is a genetic male who's chromosomally masculine. That's a bad sentence. Forget it. Chromosomally male. An X and Y chromosome. It can make testosterone, but it doesn't matter. The testosterone can't work because there's no receptor. So you can't target the cell, and the testosterone message doesn't get through. These men, these genetic males, have an external reproductive anatomy that's so much like a woman that they are raised female. They don't menstruate. When they don't menstruate, then that's usually the point at which a medical diagnosis is made, and it has determined that these people have this problem. Essentially, uniformly, these genetic males who can't respond to testosterone are raised as women and have sexual attraction to men. That's what your brain looks like from an MRI image. We focus on an organ in the brain called the hypothalamus. It's important for a whole lot of things. None of you would like to be without your hypothalamus. There's a specific part of this organ where there's a collection of nerve endings. It's called a nucleus of nerve endings. It's about the size of a grain of rice. There are a number of these. One of them has this fancy name. A number of years ago, Simon LeVay took samples of the brains of deceased person and measured the volume of this part of the hypothalamus, found that it was sexually dimorphic, and found that gay men were atypical. So this part of the brain of gay men is intermediate in its volume between heterosexual men and women. That's been repeated by William Vine in New York City. These are the data. So the volumes for straight men in black, the volumes for straight women at the right in white, and for gay men intermediate. When Simon LeVay published in Science, this drew a lot of attention from media and from people everywhere. It was criticized for its strategy, for the way in which the experiment was conducted. Many people were not ready to believe it because a number of the gay men in the study had died from AIDS. So people were asking, does AIDS cause the effect? Vine in his study was able to demonstrate that presence of HIV/AIDS has no impact on this result. This is particularly interesting to me because William Bine has been cited repeatedly by LDS social services and others in the LDS community unwilling to accept a biological explanation for homosexuality. And now it turns out that it's his laboratory that replicates the original finding of Simon LeVay. Men's brains and women's brains are not the same. Homosexual men's brains are not the same as heterosexual men's brains, and same for women. I'll cite just one study with animals. You can do important things with animals that you can't do with people. 8% of sheep, of male sheep, will not act normally in their reproductive behavior, will not act like the 92% of their brothers who are attracted to use, who will mount and ejaculate. These 8% of male attracted rams won't do that. In fact, they will allow themselves to be mounted by female attracted rams. So in studies done from populations of animals in northern Idaho and at the University of Oregon, we're going to look at the same measure that I just showed you from the human studies, and that is the size of this area of the hypothalamus. As best we can tell, it's the same in the sheep as it is in the human. There are 1, 2, 3 measures of anatomical structure: volume, number of neurons, and length. This is density. What you can see is that density doesn't show any difference, but the other three show differences. Here are the ewes on the left. The female-oriented rams are in the middle in the dark columns, and here are the, if you will, the gay male sheep. Remember aromatase, the enzyme that receives testosterone in the brain and turns it into estrogen? Okay. These are measures of the amount of that enzyme in these same animals, and the same conclusion applies. The male-oriented rams are female-like in the amount of this enzyme, and here's an actual measure of how much of the gene that encodes the aromatase enzyme is expressed. Brains are not the same structurally, brains are not the same functionally. Finally, investigators in Scandinavia have used PET scans to look at how human brains react to certain compounds that might be pheromones. Think perfumes. Think volatile materials which you can accept in the insect world are responsible for bringing males and females together. There has been a controversy, a long-standing controversy, about whether or not such things exist in humans. They probably do. So this is a map of the brain. We're looking at the hypothalamus. And there are three geometrical shapes here to represent where the brain is active as measured by PET scan photography, when human beings are exposed to two different kinds of compounds. One of them is derived from testosterone.